So now moving on to our next laureate, who is Dr. Timothy Springer, who is the Latham Family Professor of Biological Chemistry and Molecular Pharmacology, Professor of Medicine at Harvard Medical School and Boston Children's Hospital. He's also the chairman of the Institute for Protein Innovation. And he was awarded for the discovery of the first immune system adhesion molecules, elucidation of their roles in antigen recognition and leukocyte homing, and translation of these discoveries into therapeutics for autoimmune disease. And his lecture is called Basic Science on Cell Surface Receptors, Therapeutics, and the Institute for Protein Innovation.
Thanks. It's a real honor, and it's a real honor to follow Susan and Ron, who are really esteemed colleagues. And it's a real honor for my whole laboratory and my family. And I'd like to tell you about my life's journey through science today. And I started out, as one of 6 kids growing up in California. And so my 3 older siblings are with me here today. And, some of my 5 children, my wife, Jeff Finn, it's a real pleasure to share this all with him. So after 1 year at Yale, I worked as a VISTA volunteer on the Yamba Shoshone Reservation in Nevada. I helped the tribal council draft resolutions that were sent to senators. I helped them get a road paved so a bus could take students over a mountain pass in the wintertime so they didn't have to live away from home to go to high school. And I led a teenager neighborhood youth corps group installing surplus railroad ties as fence posts. And I visited those kids after they were grown up about 45 years later. And the kids were in great shape, and the fence posts were still there, as was the house where I lived. And that was really an amazing experience for a 19-year-old. So I then went to UC Berkeley, and I loved physics, organic chemistry, and wound up majoring in biochemistry with Dan Koshland, who really inspired in me a love for conformational change in proteins, which I'm working on today. And that was me then. Like Ron, I've changed a little bit since. So I did a PhD with Jack Strominger, and I worked with glycoproteins that were natively embedded in lipids that formed a plasma membrane around cells. They were very hard to extract from membranes, and one had to use detergents, and they were very hard to purify. So I leapt at the chance to work with Cesar Milstein for my postdoc, who had just discovered how to make monoclonal antibodies. And I used them as tools to break open a whole new area of the molecular basis of immune cell-cell interactions. When I got to Harvard Medical School in 1977. And in those days, antibodies were very well understood. They were magic bullets. They were secreted by B lymphocytes into the bloodstream, and they did their jobs remotely at a distance. On the other hand, T cells were very different, and they also recognized foreign antigen, but they did it by direct cell-cell contact. So here is a T cell binding to and recognizing an infected antigen-bearing cell. And we called this a conjugate at the time. And a suction cup-like contact was formed between the T cell and the target cell that we now call an immune synapse. And I was about to discover a molecule we called LFA1, which we now know forms a ring around this synapse and sort of helps create the suction, that stabilizes that contact. So at the time, the killer cells were thought to deliver the lethal hit, a knockout punch to kill the cell across the synapse. But nothing was known about the molecules involved. In fact, so little was known about the molecules that most of it was wrong. I discovered fraud on antigen-specific helper factors in Alan Monroe's lab in Cambridge before I switched to working with Cesar. And when I joined Harvard Medical School, my boss, Baruch Benassaraf, and 3 junior faculty were in the midst of a massive self-delusion. They were publishing on antigen-specific suppressor factors and suppressor cells, which were to be consigned to the trash heaps of science by the 1990s. Now, they, like other immunologists at the time, were obsessed with specificity for antigen. Some people thought that the antigen receptors were Ig-like, and some thought the T cell receptors were like major histocompatibility antigens that were the targets for transplant rejection and to which immune response genes mapped. But these were all ideas, and there were tons of papers that were published on them that were wrong in the 1970s. And so I was looking for something a little different. And let's see here. And yes, and so one hard fact we knew at the time was that this contact was absolutely requiring the presence of the divalent cation magnesium. If you didn't have magnesium, this cell-cell conjugate would not form. Now, a lot of people thought that antigen receptors were going to be like antibodies. And I certainly knew that antibodies would not require magnesium. And I also knew that other cells outside the immune system required divalent cations to bind to other cells or to basement membranes. So I reasoned that there might be some unifying principle behind this requirement for magnesium. And this unifying principle later turned out to be the integrins. So I decided to try to find this underlying principle using antibodies. And I screened for antibodies that would prevent this antigen-bearing cell from being killed by the T cell, and thereby honed in on the functionally important molecules on each side of this cell-cell interaction. And in 1981, I reported on a lymphocyte function-associated antigen called LFA1 that's required for antigen-specific recognition. And then in 1982, I also discovered LFA1 in humans and LFA2 or CD2 and LFA3. So then in 1984, the antigen receptor was cloned by a number of groups and indeed was found out to be similar in principle to antibodies. And then in 1986, we showed that LFA3 was a counterreceptor for CD2. In 1986, we also identified a counterreceptor for LFA1 called ICAM1, and that same year, we also showed that it was the interaction between LFA1 and ICAM1 that was magnesium dependent. And indeed, many years later, we crystallized a complex between these molecules and showed that the magnesium was right at the center of the interaction site and coordinated the integrin to the ICAM. In 1987, Pam Bjorkman and Don Wiley and my former PhD advisor, Jack Strominger, determined the crystal structure of the major histocompatibility complex and showed that it was bound to an antigen fragment called a peptide, and that was how the T cell receptor was able to recognize both MHC and foreign antigen at the same time. Now, immunologists found my work heretical. They were convinced that I must be wrong because the molecules I had discovered would make T cells non-specific. I used to go to Toxic Gordon conferences, and it was sort of typical, you know, when we had drinks at the evening, that somebody would pass me a napkin down the table, and it would say, "It doesn't work." You know, they just didn't get it. It was almost to this day, 30 years ago, when we showed how it worked. And that was because LFA1 amplified antigen recognition signals. What we found was that T cell receptor activation stimulated adhesiveness through LFA1. So it made antigen recognition actually more sensitive, not less sensitive, not nonspecific. And it also allowed many other cell-cell recognition molecules to be compatible with antigen-specific adhesion and to regulate immune responses. So after that time period, the tide turned, and now a whole bunch of immunologists started searching for other pairs of recognition molecules that could regulate immune responses. And so our discovery that LFA1 amplified antigen specificity provided the conceptual framework for the discovery that other cell recognition receptors could regulate immune responses without abolishing antigen specificity. And they could upregulate or downregulate immune responses. And many such pairs of receptors and counterreceptors were discovered in the 1990s, the 2000s, and up until the present decade. And these discoveries have already been recognized with Nobel Prizes to Jim Allison and Tasuku Honjo and their colleagues for the work with CTLA4 and PD-1 and PD-L1, which downregulate immune responses. And so by inhibiting those interactions with antibodies, which is another thing we pioneered, you could cure and save the lives of thousands of patients with cancer. Now, the discovery in '82 that blocking either LFA1, CD2, or LFA3 could block cell-mediated immunity also had an important implication for therapy. So it meant that blocking either one of these pathways would be sufficient to block an immune response. And one could block an immune response that went awry because the lymphocytes were overactive or specific molecules on their surface were overactive. So you could block those molecules from interacting with one another and counteract disease. And so Mike Dustin, who made that earlier discovery with me I just showed, and I purified LFA3, and we sequenced the protein and cloned the cDNA in collaboration with Biogen. And the chimera, in which the LFA3 ectodomain was fused to the FC fragment of an antibody, was approved for psoriasis in 2003. And I like the name LFA-CEPT. It was basically an antagonist of an LFA. That was the generic name. And similarly, Genentech developed an antibody to LFA1, and this antibody, Raptiva or efaolizumab, was also highly effective and approved for psoriasis in 2003. And these therapies have helped many patients. Very importantly, they also pioneered a new modality of treating disease with drugs that block cell recognition receptors. Such therapies to checkpoint inhibitors have already been recognized, as I just mentioned, with Nobel Prizes. And 2 drugs to further cell recognition receptors in the integrin family have also been approved, as we will see now. So amazingly, it's now 40 years since I published my first paper on an integrin, MAC1. And to a biochemist, it was pretty unmistakable early on that LFA1 and MAC1 looked very similarly in SDS Page. And by 1982, we had proven that they were built, they were machines that were built from 2 components, beta and alpha. And their betas were identical, and their alphas were distinct, but they were related like brothers and sisters, thus defining a protein family that is now called the integrins. And in 1983, my laboratory found another sister called alpha X beta 2. And then in 1986, other scientists working on extracellular matrix receptors started sequencing their alpha subunits and found that their sequences were homologous to the alpha subunit sequences we'd published for these years earlier. And thus, further members of the family rapidly started to appear and reached this level and continued to grow until in 1999, there were 24 different alpha beta heterodimers that were identified. And it became, and all of these receptors require magnesium for their function, and magnesium has a very important role in the ligand binding site. And thus the unifying principle for adhesiveness of all cells in the body began to take shape as the integrin family. Now, as I alluded to, integrins are very important therapeutic targets. The first drugs to be approved were FAB fragments and small molecules to the fibrinogen receptor on platelets, alpha-2b beta-3. And these drugs are used as very short-term therapeutics to prevent thrombosis. And then, as I'd already mentioned, in 2003, the antibody to LFA1 was approved for psoriasis. And our discovery that the integrin LFA1 bound a member of the Ig superfamily that was expressed on endothelium was a paradigm for 2 further integrins to which drugs were approved. One is the integrin alpha-4 beta-1 that binds to the Ig superfamily member VCAM on endothelium, and it's approved and very effective in multiple sclerosis. And integrin alpha-4 beta-7 binds to the Ig superfamily mucosal addressin cell adhesion molecule, and it is selectively expressed in the gut on mucosal tissues on endothelium, and the antibody to it selectively blocks homing of leukocytes to these tissues. And I had an important role in developing this drug through a company I founded back in 1993. But as you know, in many cases, it took many years for this drug to be approved. And it is now the most effective therapeutic for treating ulcerative colitis. And a head-to-head study comparison against Humira, which is one of the biggest selling drugs in the world. It's used for rheumatoid arthritis and other conditions on which TNF levels are high. It turned out that it's in this particular disease, it's significantly superior to Humira. So it's a widely prescribed and very important drug that grew out of the work on integrins. Now, we next discovered and clinically defined and named a disease, leukocyte adhesion deficiency. Patients have recurring life-threatening bacterial infections. And these integrins are missing because of mutations in this component of the molecular machining that's absolutely required for the function, indeed for even the expression of these molecules. So these molecules are missing on the surface of patient white blood cells, which allowed the patients to be properly identified for the first time as a collective group and be rapidly diagnosed. And now the treatment is with bone marrow transplantation, which is quite effective. Now, these patients had high levels of circulating white blood cells in their bloodstream, yet they failed to form pus. You know that when you get a cut and it gets infected, you have a big red erythema, and these patients had these big red lesions, but they didn't have any pus. And there was no pus in the middle there, which we get when we have infections, which is a sign of very high accumulation of white blood cells, which give pus its whitish yellowish color. Those are white blood cells there. So that insight of discovery of this experiment of nature showed us that the integrins we were working on must not only be important for antigen-specific recognition of T cells, and we already knew a wide variety of different lymphocytes use these receptors for recognition of other cells and many kinds of functions in the immune system. But this also taught us that they must also be required for interaction of white blood cells with endothelial cells which line blood vessels. And at the time, it was known that the first step in accumulation of leukocytes at a site of inflammation was a rolling interaction of the white blood cells along the vessel wall, followed by a pausing and firm adhesion, and then immigration out of the vessel into the tissue so a large number of cells could accumulate at a specific site and fight infection. And we started working on flow chambers, parallel wall flow chambers that would replicate the flow one saw in vivo, and we would pump purified preparations of white blood cells through, and look at the ability of those cells to bind to these purified molecules using video microscopy, just like Ron Vale showed you. And if we had ICAMs there, they didn't do anything to slow down these flowing cells. But when we added another recently discovered type of molecule, a selectin, we found that the molecules would bind to the wall of the flow chamber and roll. And then if we added a chemoattractant, that could activate the cells and simulate adhesiveness of the integrins. And so here now we're looking at rolling cells, and some cells are whooshing by, they're not bound. And pretty soon we're going to start adding a chemoattractant, and it's now reaching the position of the rolling cells. And you can see that they very quickly stop rolling and develop firm adhesion. The flow is still on here, but almost nothing is happening. And in vivo, this firm adhesion is provided by immigration, is followed by immigration out of the cells into the tissue, and the traction for that is provided by the integrins. And the key step here is actually activating the cytoskeleton, the actin cytoskeleton. That's what the chemoattractants do to the G protein coupled receptors. They activate actin polymerization, and the integrin activation is downstream of that. So that's basically what I was awarded the Gairnder for, but I want to show you what we're doing now. That is working on the molecular basis of how all this works. And we've discovered that integrins have three conformational states, which are called bent closed, extended closed, and extended open. And to visit these different states, they have to be able to flex at the knees here. That's one thing called extension. And then there's another step called headpiece opening, in which this domain, a hybrid domain, swings out. And that motion is coupled to motions within this beta I domain that result in closing up of the ligand binding site to create high affinity for a ligand. And this is one type of integrin that lack alpha I domains, and there's another type of integrin that contain alpha I domains. And here, there has to be a relay step to activate adhesiveness here, but in principle, the mechanisms are similar. And we've learned a lot about the structure of integrins from X-ray crystal structures. Most of this work has come from my laboratory, and there are crystal structures now known of, I don't know, about 7 or 8 different integrins. What I'm most proud of is that we also have structures on 2 different integrins in 2 different conformational states, the open high affinity state and the closed low affinity state. And back in 2002, we showed the presence of these 3 conformational states using electron microscopy with image averaging. And when you image thousands of particles, you get these shapes, and you can begin to see the individual domains. You see the bent integrin, the extended integrin, and this is the alpha on this side and beta on that side. And here we can see that the hybrid domain has gone from here to here, and this is the open conformation. And this is an animation of the crystal structure showing you what's happening. And we're going here from the open high affinity conformation. And this is the magnesium ion that's so important here, to which the ligand binds through an acidic group. And these 2 states are regulated by conformational change in the beta I domain, in which you can see that there's a number of compensating changes. This area moves down, and that frees up room in this area for this alpha helix to move and this metal ion to move and close up and tighten the ligand binding site. And now we're going to look at the complete ectodomain of the integrin. The integrin connects to the membrane down here, which isn't shown. And we're going to now just show you 3D what the molecule looks like. Now we're going to separate the lower legs of the integrin, and that frees up an interface with the headpiece, which now can extend, and this can then be followed by this movement out of the hybrid domain to give us the high affinity state of the integrin. So the people in the field and my own laboratory have learned a lot about integrin conformational states through the use of antibodies that were actually initially identified by their ability to activate or inhibit cellular function by binding to integrins. But it turns out that they stabilize specific conformational states, and we can use them to stabilize those states or to report on those states. And so here we're showing you again the 3 integrin conformational states with different integrin, alpha 5 beta 1, and in this case it's open conformation stabilized by binding to its ligand, fibronectin. If we add an FAB, a small antibody fragment that stabilizes the extended open conformation, we only get the open conformation. We can do that with multiple independent antibodies. Antibodies to the knees can stabilize the extended conformation because they can't bind to their epitope unless the integrin extends. So they stabilize that, and then we can get 2 states, the extended closed or extended open conformation, which you see examples of here. And then we have antibodies that inhibit integrin function, and they bind to the closed conformation. So we have the bent closed conformation, or the bent or the extended closed conformation. And we can use a combination of antibodies that stabilize extension and that stabilize closing, and then we can get the extended closed conformation. So there's really this original state, and then 4 different other combinations of states or single states that we can get. And we can measure the affinities of all of those and get a lot of information about the ligand binding affinities of each of the states. So we can solve an equation like this, and then we can calculate the affinity for ligand of each state, the probability that these states exist on cell surfaces, and hence the free energy, the relative stability of these states. And we do this with fluorescent assays, with purified molecules, or with intact cells. And from this we get what's called the energy landscape of the integrin. So these guys are lower energy. And so what the data shows is that this bent closed conformation is, for some integrins, 99.8% of the integrin on the cell surface, and these are much higher energy, so they only represent a very small percentage of the integrin in the resting state. And we've also determined that these conformations have 4,000, or this conformation has 4,000 fold higher affinity than the closed conformations. And how are the integrins activated? Well, that's what we've been working on now. And one thing we've done is to measure and calculate the free energies of these integrin conformations. And as I said, in the resting state, the extended open conformation is very high affinity, and so there's very little of the integrin in this state. We then can add adapters that would bind to the cytoplasmic domain and connect to the actin cytoskeleton. We can also add ligands, and we know exactly, given any concentration of ligand, how much ligand would be bound. So we can calculate the stability of all these states. And then if we have both the adapter and ligand-bound state, we can calculate if force was applied by the cytoskeleton, how that would change the free energy of this state, because energy equals a force times the distance. So basically, we can calculate the energies of 9 or 12 different states here. And so then we can test theories that have been proposed about how integrins get activated. And what we've found is that unlike a commonly proposed theory, if you just add a large amount of an adapter protein like talin, that is not sufficient to activate the integrin. But if you have a little bit of talin there, and you add force, then force is an ultrasensitive regulator, so with just a twofold change in the amount of force, we go from the integrin being inactive to fully active. And if we have a small force of 1.5 piconewtons, and we vary the concentration of talin, then we also get activation. So I'll bet you didn't know that I was an infomercial salesman. But basically, integrins work just like this twister fruit picker, which is activated by pulling on a rope. So just as the actin cytoskeleton pulls on the integrin and exerts a tensile force across the integrin that stabilizes this active conformation. So pulling on this rope here activates the fruit picker. So, this is how tensile force can really control a conformational change over long distance. And what we're working on now is the reaction trajectory of integrin activation. So how the kinetics of ligand binding and the kinetics of integrin conformational change occur, what their timescales are, and with those things being measured, we then can understand what is the reaction trajectory for activation. What are the steps that most frequently take us from the bent closed to extended open conformation. And the next stage of my work now is starting a new institute for protein innovation. Antibodies have been hugely important in my career, and so the new institute is creating protein tools and therapeutics to realize the promise of genomics for new medicines. And our flagship project is open source antibodies to all human and mouse proteins, and I'd like to thank a large number of people from my laboratory over the years who've really made this all possible. Science is a team sport. Thank you.